My name is Elad Katz, and I am the founder of Navigate Precision Biology. This is a spin-out from the University of Dundee in Scotland. Our spin-out is concentrated around a technology called NaviPlate. That's our way to produce 384 identical organoids inside a single plate, and we can use that for a variety of applications. We started with cancer, but we now move to chronic diseases. We make those using automated systems. This is an SBS format, which everybody in drug discovery knows how to use, and we're hoping that that is a bridge between the exciting organoid technologies that are usually done very small scale, handmade, and full drug discovery automation. So we've been using microplate readers for a long time now because that is the backbone of drug discovery. So when we do our assays, we every time try to get them through the plate reader rather than through imaging, which is potentially the go-to technique for organoids. So we do a variety of things. So for example, when we do bone microtissues, we would use the microplate reader to look at bone mineralization and quantify it using the plate reader. That takes minutes rather than hours through images. We would look at calcium oscillations in our heart organoids. We will look at kidney and lung fibrosis using specified collagen probes. And the interaction with BMG around this has been really instrumental because quite often we will take a biological assay that's never been done using microplates because we know that that would give us the most robust readout. And working around designing the right filter sets and testing them has been a really key important step in every one of our 4 organoid-based projects today. So we're using a range of PHERAstars that are already in the university. At the moment, we will buy our own when we completely spin out. The number one reason is that when we started, we were always looking to miniaturize our organoids to make the absolute smallest amount of cells per well in order to utilize as many as we can. And for that, we needed the absolutely most sensitive assays. And we found that with the PHERAstar, with the filter sets, that was giving us by far the best sensitivity. We can measure, for example, live and death of cells using that microplate reader, while when we used the other ones, we just couldn't get as far as that. And the compatibility with the NaviPlate, which is our own technology, was immediate right from the beginning, which also saved us a lot of time and hassle. So it was a winner. So we use almost exclusively fluorescence, although now we're going to probably go into polarized light, again with BMG support, because we're trying to do collagen measurements in a different way. And again, this is the type of transfer of methods that we're doing with that support, having to design our own filter set for that particular assay, which may as well has never been tried in that large format before. Our future plans are to do even more readouts, really. The assays are more or less cooked. So I expect that we will be looking to do things like, plate stackers and stuff like this, because the next plan, for example, for the bone organoids involve probably 20 to 40 plates and having a student going in and out every day is a little bit funny, but again, the robustness of the readouts is so important in that we don't worry. I know it sounds absurd, but it's actually you're not worrying too much about transferring this to the microplate is a huge benefit for us in applying what's going to go next.